so I want to make a few comments about my title. I think that if you could find a simple molecule, one you could plausibly make under primordial conditions, that both reproduced and had a phenotype, you would then have an origin for life. Anyway, that's my goal. That's what I'm going to talk about. But there are lots of other origins for life. I mean, I have a colleague in a general sense, Jack Shostak, who's put an amazing amount of work into finding something that's like a simple cell that has nucleic acids inside that replicate and that divides and so on. And that's fine if you're working on any mainline event that's required to have life as we currently know it. You're working on an origin for life, and they're all equally valid. But this is the simplest one, the simplest definition I could think of. The history of life on Earth comes to a point, and as far as I can see, that's the point it comes to. That would be the simplest thing to find. A cell is much more complicated. Okay. And Darwin realized that. So here's a quote from the first edition of the Origin of Species. Darwin realized that the history of life on Earth, if his ideas were generalized, narrows to a point. One form, one primordial form. This statement, it's interesting, I think, appears only in the first edition. He pulled it from the later editions because his wife objected to the idea that it could have been so simple and that it could have been so non-canonical in the real sense. Okay, so that's what we're by some definition, that's what we're looking for. Now, I'm going to argue that it was probably ribonucleotides. And the primary reason, it's very hard to, we're talking about 4 billion years ago, so it's very hard to gather any evidence on this point. But I think there's a strong argument from continuity. Continuity being the principle that evolution proceeds from prior forms. So there's only an incremental, each step is incremental, and what pre-existed had to look like you because you are the result of that incremental step. So it must be continuous, and what I mean by that is shown here. So here's the timeline of the Earth. Amazingly, we know when the Earth was formed to 3 significant figures. It's about 4.54 to 4.56 billion years ago, because we can date the material in asteroids and so on that were from which the Earth was formed. And rather short time after that, we have complex life at about 3.85 billion years ago with complicated metabolism. So surely the origin of life was shortly before that, maybe just before what we now call the RNA world. And so if you believe in the RNA world, you have staked a logical outpost sometime here around 4 billion years ago, and the origin has to be close to that because the Earth was, it takes a while to form continents and to have a world ocean and so on. So there's maybe there's a time of the order of 100 million years, and somewhere in there are the events we're interested in. Because that has to, by the logic I was just using, be close to the RNA world. Probably it was ribonucleotides. And so that's the assumption I'm going to use. Now, where did the ribonucleotides come from? Amazingly, this is sort of an old fact, and so some of the amazement has worn off, but amazingly, life is made out of cosmochemicals. If you look around in the solar nebula, one of the most prominent molecules you can see in space is HCN, hydrogen cyanide, okay? And an old paper, which is still wonderful to go and read, from 1962 by Joan Aurot, who's a famous Catalonian cosmochemist, I guess you would call him. Well, he isn't anymore. He was once a famous Catalonian cosmochemist. He showed that you can make adenine from 5 HCNs. You first make this wonderful looking thing, which is diaminomalleonitrile, and then you isomerize that, and you've got the imidazole ring of a purine, and then there are various ways to close the 6-membered ring, and the way that you get in highest yield in a little bit of alkali in aqueous solution with HCNs around is adenine. And in fair yield, you get adenine. But you also get guanine in lower yield. And this HCN in solution throws out black junk. And if you go and you hydrolyze the black junk mildly, you get even more adenine. In other words, one of the things you're going to need to make nucleic acids and to found your energy metabolism just literally drops out of the sky. It is hard, I think, to look at this and not see that we, who of course need lots more biochemicals, but in some sense, we lie on the chemical roots that were probable on the early Earth. Now, I am oversimplifying. That's adenine. How do you get adenosine or ATP? Now, that's a little bit harder, but in fact, you can do that just by heating adenine and ribose together. Well, how do you get ribose? Well, that's a little harder, but it's possible. So the problem is not solved, but the nature of the solution, I think, is already clear. So here are the 5 HCNs in adenine. And if you want to read a modern paper that has more of the solution in it, you can read this paper by Pauner et al. In Nature a couple of years ago. They show, at the MRC, they show that you can make pyrimidine nucleotides just from very simple cosmochemicals. So we're getting there, even though we're not there. It's hard to make, once you have nucleotides, it's hard to make RNA. So there are still steps that are not clear, but that's the direction that I think we're going to go. Okay. Now, our question is a question about where life came from, and there is a complete history of life on Earth in the big tree that we can question about this. Well, you have to know a little bit about how you get trees from sequences, not very much. The idea is that if you have some sequence that's in all cells, then that sequence will change, and if you've got one change, for example, you are closely related to the creature that has the related sequence. If you have more changes, here you have 3 changes in red, you are less directly related, and so you can make a tree which is a picture of the data, a database tree that shows the relationship between all all the creatures from whom you have sequences. And this has been done a lot. So here's a famous tree. Well, there's a modern version of a famous tree. This is the three-lobed tree that was first made by Carl Woese and George Fox. And here's the reference to that. That's 1977. By comparing ribosomal RNAs. Okay, so this is, I talk as if everything is easy, but this also was not trivial. You have to be careful because you have to have a gene that's not transferred between cells because you're not really interested in the history of the gene, you're interested in the history of the organisms. So you have to have a gene that stays within an organism and can't be transferred. And so Woese used 16S ribosomal RNA because the translation apparatus is so complicated that it doesn't engage very much in lateral gene transfer. So the history of the gene sequences, the history of ribosomal RNA sequences is the same, we hope, as the history of the organisms that possessed the genes. And immediately, as soon as Woese had data, he didn't even have whole sequences, he just had oligonucleotide positions on a map. Immediately, as soon as you start asking, well, what resembles what? He found something amazing and true that about 98% of evolutionists believe is absolutely correct, that there are three kinds of organisms on Earth that are more closely related than the distance between, the sequence distance between the groups. There's a group of eukarya, creatures with nuclei like us. In fact, right here, I can't read it. I think that says animals. So here you are. And then there's a new group of organisms that nobody had even recognized that are now called archaea. They're microbes, but they're microbes that have a different history than the bacteria, which everyone thought they resembled, and everyone thought they were, actually. So the three-lobed tree, still true. This one I got from my colleague Norm Pace. The RNA world is behind the root of this tree, which is known from analysis of an outgroup. And behind the RNA world, again, is the time that we're talking about. You have probably seen this. The point here is that there are now millions of sequences, and there's starting to be thousands of genomes even that can be compared, and that you can make a much more complex tree. Right? You very quickly get beyond the resolution of even the best digital projector. So I'm only showing a tree here with a few hundred creatures on it. And again, this is a three-lobed tree. Here are the bacteria, here are the animals, here are the archaea. So it's just like the tree before, it just has more elements. Now imagine putting millions on there. And you will see my point. Life on Earth has a single origin. There is precisely 1.0 ways to originate life on Earth. There may have been several tries, there may have been other things. They all failed. They all are inferior to the way that actually succeeded. There is one way. And that's why I think that all these people who say, well, I don't know, there may be 4 people who say, oh, so distant, how will we ever know? Horse, pucky. This is so distinctive that you will know, and I will tell you in a moment how you will know precisely that you have found the way that life started on Earth. It is the one successful way in 4 billion years. Now, this is a somewhat unusual assertion that it's going to be easy to find the origin of life, and you can, like every other thing you can say about the origin, there are people who disagree. Here are two of my colleagues from the University of Colorado in Boulder, Carol Cleland and Shelley Copley, who, well, they don't disagree really, but they wrote an amazingly fun article to read about the fact that there might be creatures from another origin still around on Earth. They called it a shadow biosphere, and there's a lot of discussion of this, but it hasn't turned up. I mean, that's my point. It has not turned up, but it's a fun thing to think about, and you can read about it here. They would have to be microbes, right? They would have to come to your attention in some way that didn't depend on grabbing their ribosomal RNA sequences, because they might not have ribosomes, and certainly their ribosomes wouldn't have the same sequences. So how will we know this one true route, the one true route to an indefinitely evolving form of life on Earth? Well, there in fact is a logical way to go about this. And now I'm going to spend some time talking about that because it's not usually talked about. So what I'm going to talk about, I'm going to attempt to talk about studying things that are out of reach. I mean, that's what we have to do with the origin of life. There are things that you can't drag into the lab and study. And all of evolution is in that category. So how do you get secure information about those things? And as an example that's worked out, much better than the origin of life is worked out, I'm going to talk about the suggestion of Lewis and Walter Alvarez that the non-avian dinosaurs were killed off by a big impactor 66 million years ago. Okay? And that also in scale as well as time is out of our experimental reach, and yet you can make an extremely compelling argument about it. And I want to present that as a model for what we're going to try and do with the origin of life. Okay, so this is now called the Cretaceous Paleogene impact. And there it is, 45 minutes after the impactor hit. Not a photograph, but a painting that was made for NASA. Okay, the means we're going to use is due to Thomas Bayes, English cleric. He published one paper in his life. After his death, he gave it to a friend. He said, "Please read this to the Royal Society." And he did. And the paper contained this amazing, subtle thing that we're now going to talk about. Okay, so don't flinch, because there are equations on here. But this is simple, and you don't have to think about you may already have thought about the equations. So here are some ideas from the foundations of probability calculus. This is a space where events happen. Areas are the probability of the events, and here are two kinds of events with an intersection, which is in both circle A and circle B. Very simple. One step from the definition of the intersection gives you this thing where the vertical bar is given that. So the probability of B, given that A has happened, is this. That's Bayes' theorem. So not yet obvious. Don't be disappointed if it's not yet obvious what that means, because I haven't said it. So now instead of A and B, we talk about having a hypothesis and getting data. The probability of your hypothesis, vertical slash, given some data, and Bayes tells you how to update your ideas. In fact, now writing it out the way we want to use it, the probability of some hypothesis given some new data is the old probability of the hypothesis times a quotient that's just the probability of the data given that the hypothesis is true over the general probability of observing the data by any means. Okay? So this says something that seems dead obvious, but it's powerful. It's powerful because it's quantitative, inevitable, and comes straight in one step from the foundations of mathematics. It has to be true. So it's better than intuition that the influence that data should have on your hypothesis is related to the probability that you observe the data given that the hypothesis is true. The increase in the probability of observing the data if your hypothesis is true. Okay, so that gives us, I argue, gives us the handle that we need. For example, Luis Alvarez and Walter Alvarez knew that all over the Earth there is a sudden transition in the rocks at 66 million years ago, and in that transitory band of just a few millimeters of stuff, there's a lot of iridium, 100 times as much iridium, the element iridium, than there is elsewhere. Iridium's a heavy metal. On Earth, it sank into the core when the planet differentiated. So there's not much iridium on the surface. There's a lot of iridium running around in bodies in the universe that have never differentiated in that sense. So when you're hit by a meteorite, there's a deposit of iridium. So they said, well, 66 million years ago, the Earth was hit by something that contained a lot of iridium, a meteorite, and because dinosaur fossils occur up to that point, non-avian dinosaur fossils occur up to that point, but never afterward. That, they said, is what killed off the dinosaurs. So they didn't say it, but they were using the Bayes logic. The probability that an impact killed the dinosaurs, given the iridium, is the general probability of impacts times the probability that the iridium is on the surface given an impact over the low probability of iridium, because it's all gone into the core. So the iridium spike at 66 million years is evidence for the impact. Now there's 30 years of discussion, and the point is that Bayes tells you what to do about different types of experiments. So usefully, if you have two different kinds of experimental data and you have your original hypothesis, each time you get things that are better explained, like the iridium, by having an impact than by just general events, you multiply the original probability you started with. So if you do experiments that elevate the probability of your hypothesis tenfold, and you do 2 of them, you've elevated the probability 100-fold. You can just arbitrarily elevate the probability of some hypothesis, however improbable it starts out as. You eventually get a compelling argument. That is to say, this kind of indirect work proves hypotheses in the same sense that normal experiments do, by elevating the probability of something till you finally say uncle and start to believe it. And there were all kinds of evidence, because people hated this idea that life on Earth was subject to the apparent hazard of a catastrophe that was potent enough to wipe out 50 to 75% of the species on Earth. Not just the non-avian dinos, but 50 to 75% of the creatures on Earth are gone at the Cretaceous Paleogene transition, as it's now called. Well, okay, so there are lots of other kinds of evidence. There are tektites. There are these glassy things that are the result of melting the rock at the point where the bolide hits. And there are these decrease in frequency as you go out away from the point of impact. So all around the Caribbean, there are these waves of glassy, melted stuff. There's shocked quartz. So this is a quartz crystal under cross polarizers. And you see these striations. That's where the crystal lattice has just been dislocated by immense pressure. And once again, there's this stuff all over the Caribbean. In fact, in a way, the most improbable data, the probability of the data with the most impressive increase in probability, given that there was an impact, is the crater itself, which has been found. So this is Yucatan. The Gulf of Mexico is out here. This is Yucatan. In northern Yucatan, there's a crater half, I shouldn't be pointing at that screen, right? So there's a crater half on and half off land, sort of. And here there's marine sand that has been moved by an immense tidal wave in the past. And then there's the crater itself sitting there around the town of Chicxulub. So so this 180 kilometer crater, one of the largest impact features on Earth, actually is the final element in the argument I'm going to talk about. And I can't resist showing you this, which really makes my hair stand on end. This is by mapping the gravity and magnetic anomalies over the coast of Yucatan. So the coast of Yucatan goes right through here, as in our previous map. You can see the impactor coming in. You can see the crater. And you can see a plume of ejecta going out northwest toward the United States. Some of which I showed you when we looked at that spike of those rocks with the spike of iridium in Colorado. An amazing record of the event. So you know that that baby came in at a low angle and crashed at this point, tossing stuff to the northwest. Okay, so once again, there's a lot of disagreement and discussion. Okay, people didn't want to believe it. There's a lot of discussion of the fact that about this time, 66 million years ago, there's a huge volcanic eruption in India, in northwest India. The residue today is called the Deccan Traps, and so a lot of people wanted to test the hypothesis that it was volcanism on a huge scale that killed the dinosaurs. But in fact, the whole thing has been studied rather recently, and you can read about it here in Science in 2010. You can read about the conclusion here. The extinctions in time, there is no exception to the extinctions coinciding with the iridium layer, and the iridium layer is dated very precisely. So it was, in fact, the bolide that killed the dinosaurs and everybody else. And it's, in fact, the fact that these things come together that is so convincing. So here's my attempt to illustrate this. So here's a Rex out for an afternoon walk. He is roaring at the incoming 10-kilometer meteorite, which is about to change his life forever. And the fact that you can see this as a picture in which the pieces fit together, so that you have much greater confidence in the overall picture, you have much greater confidence that this fits because these other things make the site for it. That's the multiplicative character of independent evidence, an extremely powerful way of gathering evidence and applying it to a single hypothesis. Now, it's this kind of Bayesian convergence that I argue, and I'm going to talk about some evidence next time, can be similarly applied to grasp something about the origin of life, even though it's even more out of reach than the extinction of the dinosaurs. And I guess I should stop there. Oh, I have 4 minutes? Well, let's see. Okay. So what I'm going to do next is show you now. So this is I don't know. This is general background. Next starts the evidence. So the first item of evidence I want to talk about is how hard really is it to do primordial chemistry? I mean, there's all this, you can't depend on the amounts of stuff, and you can't depend on the time that it's going to be made, and the amounts vary, and everything's unstable. So the next topic is any of that a barrier to something you'd like to do, like make a nucleic acid? And the answer is no. The answer is no. Your intuition completely misleads you. And I'll make that argument to you to start out next time. Then, on that basis that you could have done nucleic acid chemistry primordially, my lab went out to look for nucleic acids which might plausibly have replicated. And to do that, they have to be tiny. So are there tiny things that can replicate? That's the novelty of this. No one had a tiny thing that could replicate or reproduce somehow, and that also could have a chemical phenotype, so that you have the material for evolution, right? You have something that reproduces, that changes its environment, that does chemical reactions, that is a catalyst, and we have a candidate for that role. And I'm going to argue it's actually associated already with a Bayesian convergence on the hypothesis that small ribonucleotides originated life. And so that's what we'll do next time, I hope.